All right, our last objective for blood, talking about formed elements of blood. So we have a couple of objectives here. We want to define hemopoiesis and explain the role of colony-stimulating factors, describe the 4 cellular stages of erythropoiesis, compare the production of granulocytes, monocytes, and lymphocytes in leukopoiesis, summarize the process by which platelets are formed in thrombopoiesis, describe the structure of erythrocytes, and list the events by which erythrocyte production is stimulated. Our next set of objectives are to explain the process by which erythrocyte components are recycled in the body, compare and contrast the different blood types and their importance when transfusing blood, explain the main function of leukocytes, distinguish between granulocytes and agranulocytes, and compare and contrast the various types, explain what is meant by a differential count and how it is clinically useful, and explain the structure and function of platelets. All right, so starting off, our hemopoiesis is just the production of formed elements. So it occurs in red bone marrow of certain bones, for example, in the femur. Hemocytoblasts are stem cells. They're pluripotent, meaning they can differentiate into many types of cells, and they produce 2 different lines, the myeloid line and the lymphoid line. So the myeloid line forms erythrocytes, or red blood cells, all leukocytes except for lymphocytes, and megakaryocytes. These are cells that are going to produce platelets. And then we have our lymphoid line, which forms only our lymphocytes. So with hemopoiesis, we have these CSFs, or colony stimulating factors, which are going to stimulate hemopoiesis. So we have 6 different types. The first one is the multi-colony stimulating factor. This increases the formation of erythrocytes, granulocytes, monocytes, and platelets. Then we have our granulocyte macrophage colony stimulating factor, which accelerates formation of granulocytes and monocytes. We have our granulocyte colony stimulating factor, which just stimulates the formation of granulocytes. Notice the trend, right? They tell us what they're stimulating. The macrophage colony stimulating factor, of course, again, is going to be those production of monocytes. Thrombopoietin is going to stimulate the production of platelets. And erythropoietin, or EPO, is a hormone from the kidneys that are going to increase the rate of production and maturation of our erythrocytes. So talking about erythropoiesis, this is just red blood cell production. So actually producing red blood cells requires a couple different things, specifically iron, B vitamins, and amino acids. So if you ever hear of people who are vegetarian lacking B vitamins, and they may go ahead and have a lower red blood count, that's why. So it begins with myeloid stem cells, and these respond to that multi-colony stimulating factor, or CSF, and it forms a progenitor cell. From there, it's going to form a proerythroblast, which is a large nucleated cell. It's going to turn into what's called an erythroblast. Remember, blast just means a bud or a germinating cell. It's going to be smaller now and produces hemoglobin. And then it's going to become a normoblast, which is still smaller and has more hemoglobin and is also without a nucleus, or anucleate. It becomes next a reticulocyte, which lacks organelles except for ribosomes that make hemoglobin. So notice red blood cells are just slowly, not deteriorating, but becoming smaller and just filled with hemoglobin. And then it's going to turn into an erythrocyte, where the ribosomes have completely degenerated and it's just a bunch of hemoglobin. All right, so leukopoiesis is the production again of leukocytes. It involves the maturation of granulocytes, monocytes, and lymphocytes. So granulocytes are neutrophils, basophils, and eosinophils. The multi-CSF, or colony stimulating factor, and GM colony stimulating factor cause myeloid stem cells to form progenitor cells. The progenitor cells then become a myeloblast that becomes a granulocyte. So not as many steps as in erythropoiesis. Monocytes also are derived from myeloid stem cells, and the stem cell differentiates again into a progenitor cell, which then is going to be prompted to become a monoblast from MCSF. And then the monoblast becomes a promonocyte, which matures into a monocyte. And our last one here for leukopoiesis is that lymphocytes are derived from lymphoid stem cells. The stem cells differentiate into two different types, the B lymphoblasts and the T lymphoblasts. Lymphoblasts are going to mature then into the B lymphocytes and the T lymphocytes, and some lymphoid stem cells are going to differentiate directly into natural killer cells. So next we have our thrombopoiesis, which is just platelet production. And we have these cells that are megakaryoblast, which are produced from the myeloid stem cell. They're going to form a megakaryocyte under influence of thrombopoietin, and it has a pretty large size and a multi-lobe nucleus. The megakaryocyte produces thousands of platelets, and so these large cells are going to produce proplatelets, or long extensions from themselves, and they extend through blood vessel walls into the bloodstream. And then from there, the blood flow, as it goes through, is going to slice off these little fragments from the megakaryocyte, which are going to become platelets, which is why most of the time when we think about platelets, they're just pieces of a cell, and that's even how we're describing it, because that's literally what it is. So this picture is just going to show us, of course, that hemopoiesis, starting out with a hemocytoblast, which is going to turn into either the myeloid line, coming down this way, right, or into the lympho line, which is this one here, which is only going to produce those little B lymphoblasts and T lymphoblasts, and then eventually become a B lymphocyte, T lymphocyte, and a natural killer cell. And then, of course, we can see how the erythrocytes are built, starting out big as a progenitor cell, and then slowly just losing the nucleus, losing the organelles, and then eventually just being filled up with hemoglobin and becoming an erythrocyte. All right, and this is another awesome picture to show, where we might go ahead, and this is the humerus, the head of the humerus. We find that red bone marrow going to be having megakaryocytes, so showing our thrombopoiesis, and the formation of platelets here, those little arms coming out from the megakaryocyte, and those little platelets coming off. Pretty cool, actually. All right. So red blood cells, let's talk a little bit about them. They're small, flexible formed elements, and they lack a nucleus and cellular organs, but they're packed with hemoglobin. So they have a bioconcave disc structure, which has latticework of spectrin protein providing support and flexibility. So they can stack and line up in single file, called a rouleau, which is what we can see here in this picture, which is just cool. They transport oxygen and carbon dioxide between the tissues and lungs, and the way they can do that is because of the fact they're just absolutely packed with this hemoglobin protein. So hemoglobin is a red pigmented protein, hence the color of our blood. And again, it's made for transporting the gases oxygen and carbon dioxide. Blood is going to be, so the hemoglobin is going to be considered oxygenated when it's maximally loaded with oxygen, and deoxygenated when some of the oxygen is lost. So each hemoglobin molecule is composed of 4 globins. There are 2 alpha chains and 2 beta chains. Each chain has what's called a heme group on it, which is a porphyrin ring with an iron ion in its center. Oxygen is really attracted to iron, so it's going to bind to that iron ion so that each hemoglobin can bind 4 oxygen molecules. So oxygen binds to the iron. It's fairly weak, which is good, right? It's attracted to it compared to everything else in the erythrocyte, but we don't want it to be so attracted that it won't release when it goes ahead and goes to the rest of the body tissues after rapidly attaching in the lungs and then being able to rapidly detach in the body tissues. So carbon dioxide also binds to the protein, but it specifically binds to the globin protein instead of the iron. And again, the binding is fairly weak, and so it can attach in the body tissues and then detach in the lungs. And so here's just the structure of hemoglobin, and you can see those 4 globin chains, the 2 alpha and the 2 beta chains, and then that little heme group where our oxygen is going to bind. And then I'm going to stop here and make another video for you to watch after this.